Cell-free protein synthesis is an emerging technology in systems and synthetic biology for the in vitro production of proteins. Cell-free protein synthesis systems have no cell wall, thus we have direct access to metabolites. In this protocol, we quantify the absolute measurements of 40 metabolites involved in central carbon and energy metabolism, which allows us to characterize cell-free metabolism.
The protocol relies on derivatizing the compounds with aniline, which allows for better separation and higher mass spectrometry resolution. In addition, we use internal standards with an isotopically labeled aniline for absolute quantification of the metabolites. The tagged metabolites co-elute, which eliminates the effects of ion suppression, allowing for the accurate quantification of the compounds.
Working in a hood, combine 550 microliters of the aniline with 337.5 microliters of LC-MS grade water and 112.5 microliters of 12 molar hydrochloric acid in a centrifuge tube. Vortex well and store the 6 molar aniline solution at pH 4.5 and 4 degrees Celsius. To prepare a 6 molar carbon-13 aniline solution at pH 4.5, combine 250 milligrams of carbon-13 aniline with 132 microliters of water and 44 microliters of 12 molar hydrochloric acid. Vortex well and store at 4 degrees Celsius. To prepare 200 milligrams